Gloria Regina is full professor at the University Federal of Minas Gerais, Brazil, and leader of a research group in the areas of molecular biology and bioinformatics, acted as head of the bioinformatics graduate program at the University Federal of Minas Gerais. Am I right with that? And as a president of the Brazilian Association of Bioinformatics and Computational Biology. This is the ABC. I must say that it's a huge association. I was in some meetings and they do very nice meetings. So she works in the fields of genomic, sequencing, assembling, and annotation of bacteria and yeast genomes, and transcriptomic, that include RNA sequencing, analysis of protozoa, flatworm, mouse, and human. And her more recent focus is on understanding the mechanism and underlying the expression of different transcript isoforms for protein coding and non-coding genes in normal and stressful conditions as disease and drug treatment in human and mouse. I must say that Gloria Regina is a referent in all those areas with a huge experience of background and knowledge. So enjoy her talk. I leave you with her.
Well, Cristina, thank you so much for your very kind introduction. It's a pleasure, enormous pleasure to be here. I'd like to thank you and the other organizers for this wonderful meeting. As I said this morning, it's not easy to organize a meeting like this, international meeting, in the middle of a pandemic, and you're doing great job. And I was present in some sessions yesterday and today in the morning, and the works are excellent. Congratulations to you and to the other speakers as well. Thank you. I will share my screen. Oops. Is it okay?
It is okay, but it's not full.
It's not full.
Yes, it looks okay to me.
It looks okay. Okay.
Yes.
Now it's a full screen. Is it a full screen now?
It is. It's perfect.
Okay. Great. I ask you to advise me when I have like 10 and 5 minutes, because for me it's very difficult to keep in time.
Okay.
I really appreciate that because I usually like go ahead and forget about the time. Okay.
I will tell you.
My presentation today will be about alternative splicing, modulating the cell response to SARS-CoV-2 infection and the new coronavirus. And this is not genomics, but it's transcriptomics. The session is about genomics, but it's a mixture of genomics and transcriptomics. And it's an interesting topic I am working recently. And here we see a picture of the alternative splicing. And the splicing is a central process in the regulation of gene expression in eukaryotes, and is very conserved and highly conserved in different organisms. Since yeast to man, it's the process is very conserved, and all the proteins involved in RNA. And it's a critical process to regulate the gene expression post-transcriptionally, and it provides an expansion of the proteome, as you can see here. In this case, you can skip some exons during the alternative splicing process, and you can have slightly different proteins in the end. And this is why you can expand the proteome in eukaryotic organisms. And you can also maintain the introns and give different proteins as well. It's very interesting because it's an important process in humans. 100% of the human genes produce at least 2 alternative mRNAs, isoforms. And then splicing is absolutely important. And it's a process you can control in disease, maybe as a new drug for a kind of disease. And here we have a classical signal, effectors, and regulators of RNA splicing in humans. It's done by, the kind of introns we have are called introns from the group 2. And the splicing is made by a machinery called spliceosome. And the spliceosome is composed of proteins and small RNAs called small nuclear RNAs, such as U1, U2, U4, U5, U6, and U2AF. That is a different complex. All of these are part of the major spliceosome, and there are other factors that are really important for the process, especially the alternative splicing, that are heterogeneous nuclear ribonucleoproteins and serine, arginine-rich repeated proteins. In the RNA, there are many features that are important for splicing. And here you see the exon 1, the exon 2, and the intron in the middle. For the intron, you have features as the donor site. It's composed by different nucleotides, but it's very conserved in the beginning by the GU nucleotides. And this is the acceptor site, the splicing site. It finishes with an AG, and there are other nucleotides that are conserved, especially a region that is rich in pyrimidines. It's called polypyrimidine tract. And another feature important is the A, is a nucleotide, adenine nucleotide in this region that is called branch point. And then in order for the spliceosome to do the splicing, it's necessary the recognition of the acceptor site for the splicing, the donor site, the acceptor site, the branch point, and the polypyrimidine. And then the intron is eliminated as an lariat intermediate. Besides these very important features you see in the intron, there are other features you see in the exons and in the introns, like the ESS, the exonic splicing silencer, or intronic splicing silencer, or intronic splicing enhancer and exonic splicing enhancer. These regions are conserved and are recognized by, the enhancers are recognized by the SR proteins, and the silencers are recognized by HN RNPs. One point that is very interesting is that spliceosome is an attractive new target for small molecule binding, antisense oligonucleotides, and genomic editing therapeutic for interventions against different kinds of disease. There are different patterns of splicing, the basic ones and the complex alternative splicing patterns. The basic ones, and probably the most important, is called exon skipping, when you lose the exon and you link the exons, the neighbor's exons, or the intron retention. The intron is not recognized as an intron and is maintained in the final and mature RNA. There are other kinds of basic alternative splicing patterns that uses alternative exons. They are called mutually exclusive exons. If you have this, you don't have this one, and vice versa. You can use alternative splicing site like donors, alternative donors, or alternative receptors, and then you will not have the whole exon, but part of the exon, the 5' exon or 3' exon. And you can also have alternative first exons. And then the promoter can start from here or from here, and then the pattern of splicing is completely different. Just to emphasize that the dark blues are called constitutively spliced exons, and the red and the light blue are, or green, are alternative spliced exons. And there are other very, very complex patterns that give rise to different isoforms of alternative transcripts. Sometimes I will call isoforms, sometimes I will call them biotypes. It's the same thing, isoforms of transcripts or biotypes of transcripts. And here you can see how this is regulated at the level of the pre-mRNA, and the after the transcription is processing and producing the primary RNA. All these factors bind to the elements in the intron and in the exon, and they can control if the exon will be maintained, removed, partially maintained, partially removed. And these proteins, these factors, can open positively or negatively in this the machinery or the components of the spliceosome. Retained introns are very important for mammalians, and especially for humans. There is an increase in the number of transcripts that retain introns in humans. And what happens when an intron is retained in the transcript? The problem is sometimes it changes the frame, or it causes the frame shift in the ribosomal, during the ribosomal translation, and produces a completely different protein. And that's why it's so dangerous to keep an intron in a mature transcript. But sometimes, especially when you have very, very long introns, it's difficult to recognize the patterns, the features that are in the intron, and sometimes part of the intron is retained, the part or the whole intron is retained. I have to open a parenthesis here to say that in humans, introns are much, much larger than the exons. The average size of an exon in human genes is close to 200 nucleotides or base pair. And for introns is more than 5,000 base pairs. And then the introns are very long, the exons are very short, and you have many genes with multiple exons and introns. Very rare genes with just one exon that we call monoexonic genes. They are very, very rare in the human genome. If you keep the intron during the splicing, sometimes the RNA is kept in the nucleus, and then it's degraded in the nucleus, and it's not exported to the cytoplasm, and then this controls the levels of this transcript available for translation. It's a kind of fine-tune in the regulation of gene expression. Sometimes some transcripts that retain introns go to the cytoplasm, and they contain introns, especially close to the 5' end of the transcript. Sometimes it's the first intron or very close to the 5' end. And the intron has a premature stop codon, and then he is PTC. And when this transcript goes to the cytoplasm, arrives there, it's degraded by a very efficient and conserved mechanism called nonsense mediated decay, because this mechanism is important to remove wrong transcripts that underwent bad splicing. But sometimes when the intron is more in the middle of the transcript, the mRNA, or more in the end, it's impossible to be recognized by the nonsense mediated decay pathway. And the intron is retained, and sometimes it continues the same frame for translation. And that's, it generates a new protein. Is it okay? It produces a new protein that carries a different part in the protein and can have even a different function or localization in the cell. And in the other cases, it produces a truncated protein because sometimes it has a stop codon in the middle or produces, there is a frame shift when you retain the intron, and it produces a protein that can do partially the same function as the other one, the correct protein, but it cannot have the full characteristics of the protein and act as a dominant negative protein. That's very interesting. This is a way to control gene expression, and it's very, very fine tuning of the gene expression in mammals, especially in primates. For this presentation, I'd like to say some words about the new concept of the transcriptome in eukaryotes. The old concept of the transcriptome says that we have genes, and we have genes that are interrupted by introns, and when you have a transcription of this gene and splicing, you generate the protein. And most of this old transcriptome concept was based in coding, protein coding genes. And then you have protein coding genes in this sense, and the sense strand and the anti-sense strand. But after we developed much better ways to analyze transcriptome using technologies of next generation sequencing and the RNA-Seq, we can now understand how different is the concept of the transcriptome, because you have a very deep look into the kinds of transcripts we produce. And probably 90% of the human genome is transcribed during the development or in different cells. And you have a bunch of different transcripts in the cells. Most of them, we have no idea what is the function, what they are doing. And most of them are consist of no coding RNAs. And then in this case, for the new concept, you have here the same gene region with 3 exons, but now we discovered that in the middle of the last exon, there is another gene. And this gene is transcribed in the antisense strand, generating an antisense no coding RNA. Sometimes you have another gene inside of an intron, especially because the introns in humans are very, very long. And then it's easy to have a gene inside of a gene. And then you have an intron that carries a new gene that is an intronic RNA, no coding RNA. And there are other genes that were localized in regions that we, in the old times, we didn't see any gene because there is no characteristic for a protein coding gene in this region, but now we know they are transcribed and they produce an intergenic no-coding RNA. Usually, these regions produce intergenic long no-coding RNAs. They can be as long no-coding RNAs can be in the intergenic region or inside of intronic, and then they are called intronic long intergenic no-coding RNA. And interestingly, this intergenic or intronic no-coding RNA, long no-coding RNA, they have exons and introns. They can be processed by splicing, and they are transcribed by the same RNA polymerase as the mRNA we know. They have cap, they have poly A tails, they have intron, and they suffer the process of splicing, but they don't code for proteins. That is very interesting. They have many other functions that I would like to say in here. They can stay in the nucleus and act as a transcriptional activator. They bind to transcription factors. They can be a repressor that helps repressor proteins or transcription factors to be removed from the promoters of enhancer. They can guide complexes to the starting site of transcription. They can help in the scaffolding of the complex that modify chromatin, and they can change chromatin status. They can act post-transcriptionally. They can be circular or linear, and they can regulate splicing in the nucleus, but in the cytoplasm, they can bind microRNA and sequester. They can be sponges of microRNA and control the levels of microRNAs. And then we know that microRNAs can be used to block translation. And then when the long no-coding RNA controls the level of the microRNA, different kinds of microRNA, they interfere with the translational rate of an mRNA. The thing that is very interesting to emphasize is why we had no idea about these long no-coding RNAs in the past. The thing is, they are very, very low expressed. Here you can see seeing here the low read analysis and the high read analysis with the deep transcriptome. And this is a protein coding gene in green, and this is the long no coding from this region. In comparison to the protein coding gene, the expression of the long no code is very, very, very low. That's why when you use different strategies that were not deep enough, we could not see these molecules in the transcriptome. And they are more tissue specific than coding RNAs. And then you see different protein coding transcripts expressed in different tissues, and the long no coding RNAs are very restricted in their expression to different tissues. And they can be even a mark for tissue specificity and can be used as a biomarker in some disease. And they, especially the circular RNAs, can go to this blood and can be analyzed as a new biomarker. They can be amplified from plasma of the organisms. Well, our focus is in the new coronavirus, the SARS-CoV-2. It causes the disease we know, COVID-19, that started last year in China. And this coronavirus is similar to other coronavirus. They have especially an important protein that's called spike protein that makes it similar to the other coronavirus. That's why it's called corona. like a coroa in Latin. And this protein, this spike protein, is recognized by a receptor in the cells called ACE2. The ACE2 is a protein important in the regulation of the different process of blood pressure in the organism. And it's recognized, it binds to the ACE2, and after that it's processed and the virus enter to the cells. The ACE2 is spread and it's expressed in different tissues and cells in the organism. For the infection with SARS-CoV, you have sometimes only local symptoms or no symptoms. Some people are asymptomatic, and local symptoms are from this respiratory upper and respiratory tract, or sometimes in the lungs, but it's not very, very severe. But in some cases, the spread of the virus in the lungs and through the blood system can cause a very, very severe disease called systemic inflammatory response syndrome. That depends on the cells that are produced, the inflammatory cells that are produced by bone marrow and other proteins produced by the liver that accumulate lead to the accumulation of prothrombotic factors and result in blood clots and thrombosis. And many cells from the immune system that are liberated from different lymphoid organs in the circulation can produce many inflammatory and cytokines, leading to what we know cytokine storm that is very hard and can cause multiple organ failure. And locally, what happens in the lungs?
Sorry,
Yes.
You have a bit more, little more than 5 minutes left if we want to leave some time for questions.
Sorry,
you have around
I
5
didn't have 30 minutes. It was not 30 minutes.
You are till 15:10. Your talk, we starting 5 minutes later will be till 15:15.
Okay, and then I have more 10 minutes, right?
Okay, yes.
Okay, great. And then I'll skip this just to say that,
sorry, nice
no problem, no problem. I used to say, I used to speak a lot. And then many different analyses of transcriptomics from cells, immune cells, say that the organism is trying to produce many different cells from innate response, immune response, or adaptive immune response. And you see in severe cases here of SARS-CoV, COVID, you have more macrophage and neutrophils in lungs. And in more moderate cases, you have more T cells, NK cells, and B cells in lungs, because this is a bronchoalveolar analysis, transcriptome analysis. And the other thing is you have also many different cytokines, especially inflammatory cytokines, such as IL-6, IL-1 beta, and CCL8. And last production that is very interesting of interferon. And interferon is a very important response against viruses. And then this is type 1 interferon and type 3 interferon. You see that the levels of these interferons are not very high in cells of lungs or different cells in the organism. Then there is a hypothesis that raised by Akiko Iwasaki that says that the response, the interferon response depends on the viral load. If you have a low viral load, you have time to produce a good interferon response. But if you have a high viral load, you have a delay in the interferon response. And then you have persistence of the virus, inflammation, and severe disease. And in this case, you have a mild disease. And sometimes this happens in the elderly people that is complicated by other comorbidities. This hypothesis says that some viral proteins are interfering with the transduction or the signaling pathways important in the response of the cell. This is a pathway for cytokine production, and this is for interferon production. And some proteins of the virus can interact directly with these proteins and can mediate or interfere with this process, not only to produce interferon, but the neighbor cells to sense the interferon. And that's why there is a problem in the amount of interferon produced and the way the cells sense the interferon and produce the genes that are stimulated by interferon. And then we use it in our work, some data from transcriptomics of the pulmonary cells, is a cell line, and also an integrative analysis using translatome, protein-protein interaction, using virus proteins, SARS-CoV-2 proteins expressed in human cells, and what kind of proteins they are interacting. This is our pipeline. It's based not only in gene expression, but in transcript expression or isotypes of transcripts that are expressed. It's not easy to have this pipeline. I don't know any paper that is very detailed in this kind of transcript focused analysis. And then in the end, we have here the types of different transcripts from each gene. Sometimes you don't have the gene that is differentially expressed, but isoforms of transcripts that are differentially expressed. And our volcano plots are based on transcripts, not only on genes. And the functional enrichment pathways or process are based on the kinds of different transcripts are produced. Our data our analyses were from public data, from Daniel Blanco Melo, and they used these cells, the alveolar cells, with different multiplicity of infections, different viral loads, a cell, wild type cell, and a cell overexpressing the receptor for SARS-CoV-2, the ACE2, at different viral load. And then we did all the analysis, the pathways analysis, and the long non-coding RNA as well. Here is just the analysis on the level of the gene. And we see that no matter the type of the cell, if it's not expressing or overexpressing ACE2, you have a very strong inflammatory response. But when you have a higher load, viral load, you have a more inflammatory response. But concerning interferon and all the genes that are sensitive to the virus, we don't have, you don't see usually expression or enrichment in the process of interferon response, depending on the amount of viral load. If you have a very low viral load, you produce interferon. But if you have a mild viral load, you don't have any enrichment in this process. But if you have a very, very high load of the virus, you still have this process enriched. And the other interesting thing is you have a downregulation of other processes, especially splicing. Splicing is one process very important to control other processes, and it's downregulated in infection. And then we quantified different types of transcripts, and we see that we have differences in the expression for protein coding genes, but other different isoforms of transcripts like retained intron, processed transcripts, etc. And here, just an example to see the gene is not differentially expressed, but the isoforms of the genes are differentially expressed. There are many examples, and these are genes involved in signaling pathways or presentation of antigens. And you see here, some isoforms are nonsense derived from nonsense mediated decay, retained introns. They are differentially expressed, but the gene is not differentially expressed. And then when you see at the level of the isoform, you see a different picture. The inflammatory response is not that high for no-coding biotypes. The no-coding biotypes will be retained intro, processed transcripts, nonsense mediated decay. But for the protein coding, it resembles the same response as we see for the gene. But when you see the biotypes for no-coding proteins, they have a different characteristics in expression, and the splicing processes are overexpressed for no-coding isotypes. And we can see this for interferon responses. And I think the most important thing in this slide is this here. This is processes related to antigen presentation. And this is response to interferon. What we see here, this are the isoforms coding for proteins. And in blue, in blue are the isoforms coding for proteins. And in red, we have no coding isoforms of the same gene. And then we see that for interferon response, we have an increase in no coding isoforms. When you have here in these two lanes, we have here cells that are overexpressing ACE2. Here, cells overexpressing ACE2, and this is wild type cells. And then more we have viruses entering in the cells, more you can differentially control the types of the isoforms that are produced. And that's depend on the pathway. And for the presentation of antigens, we have an increase in no-coding isoforms as well as for interferon response, but not for other kinds of process, such as chemokines and cytokines production. Most of the isoforms are coding. And you see the same for splicing. For splicing, you have an increase in no-coding isoforms of the proteins. We have also an increase in the long no-coding RNAs that are expressed. They are upregulated. Many of them are downregulated. And the long no-coding RNAs, the expression depends on the viral load. More viral load, more long no-coding RNAs are overexpressed, and they are related to different processes and the same process as we see in no-coding isoforms: splicing, transcription, translation, inflammatory response, energy metabolism. There are many other cases that virus controlling the expression of no-coding RNAs in humans. I'd like to skip this. And the last slide is when you have the importance of different kinds of viral infections controlling and altering the splicing machinery. They can control directly, just interacting with protein of the machinery, of splicing machinery, promoting changes in alternative splicing, or indirectly by controlling cells of the immune response or process that damage the cell. And then this is very important. Maybe is one way the virus is regulating the process, the basic process in the cells. I'd like to thank the team that was involved in this work. Is still unpublished data. And it's the leader of this research is Tomás Lucha, that is defending his PhD thesis with the help of Isabella, Professor Eder Nakaya, Rafael Polidoro, our collaborator from Indiana University, Nayara Toledo, a PhD student, Lúcio Toledo, PhD student, and Ícaro Castro, a PhD student as well. Thank you very much. I'm open for questions. I'm happy to take questions. Thank you. I'm a little bit late.
Don't worry. It was so amazing. I'm really overwhelmed with the amount of results and so interesting results you have. How did you have the time to do that? It's very new, SARS-CoV-2.
Thank you. Sorry, I usually speak too much. That's my problem. Maybe it's a bias as a professor. Sorry.
No, but it was so nice. We could be hearing you for 3 hours. So
thank you.
It raises me many questions. I haven't think about this transcript splicing regulatory system is amazing. It's so exciting, but I will ask you other people question. Here, Yesid Cuesta is asking, work Gloria, according to your pipeline, what is going to be the rule of the protein-protein interaction data sets? Do you have any hypothesis to test related with isoforms and protein-protein interactions?
Yeah, great question. We don't have the same protein data for the cells we use it for transcriptome. We have, I didn't have time to show, but we have protein-protein interactions using another cell. And of course, another viral load, but we can see many modules of interaction when the central of the module is protein from COVID. And the modules are related to interaction with proteins involved in splicing. And the other thing is we couldn't see yet, because it needs to be done, what isoforms in proteins are related to this interaction with viruses. Recently, it was a published paper saying that ACE2 has different isoforms and can be important for interaction with SARS-CoV-2. And then maybe it can be controlling the way the virus enter in the cells, or sometimes the way the virus interact with the whole cell machinery and induces the severity of the disease. We don't know yet, because it needs a much deeper proteome analysis.
Thank you, Gloria. There are too many questions. I think I have one or two more, and after that, if you want, you can stay at the chat and answer.
I'll be here. I'll be here. I can be open to answer questions by email. That's no problem. I'll be here.
So another question is, could you explain in better detail the relationship between viral load and number of long non-coding RNA expressed?
We saw that in the pulmonary cells, the epithelial pulmonary cells. And you see that when you have a very low viral load, there is no, we could not capture any change in the long no-coding RNA expression. We just started to see differences in the expression of long no-coding RNAs when we have a high viral load in wild type cells, or in the cells overexpressing ACE2. And in the cells overexpressing ACE2, the number of differentially expressed long no-coding RNAs in low viral load was much less than the number in the higher load. And then higher or the amount of virus that infects the cell, more long no-coding RNAs are differentially expressed.
Amazing. I'll make you the last question. There are many others, so please be in contact with Gloria. Yara Sousa says, amazing work. Professor Gloria, I am your fan.
Thank you. Thank you.
How do you think transcriptome annotation, especially for long non-coding RNA and other non-coding RNA, is going to change with long read sequencing?
That's very good question. Well, the problem, we have short reads and I have to reconstruct the transcript by mapping to the genome or de novo assembly of the transcriptomes. And sometimes we have mistakes, of course. But now we have a sequence of isoforms directly from PacBio or Minayo. And then we have the completely mounted, the assembled isoform. It's much more it's more feasible to be analyzed as an isoform. And then we're going to see differences in the amounts of isoform much more we can see using short reads and assembly of the novel transcriptome. And the other thing is when you use Minayo, you can do a direct sequence of the RNA. And then you can see changes like methylation in the bases, modifications in the bases. And then it's not only the type of isoform, but what kind of modification the RNA have.
Even another level of complex.
Another level. It's called epitranscriptomics. Epitranscriptomics.
Wow.
It's going to change the rate the RNA is translated, the rate the RNA is degraded.
So, thank you very much, Gloria.
It's a pleasure. It was a pleasure.
Thank you, but please be in contact with her. Thank you again very much for being here. Great talk. Great person.
Thank you. I'll be here for the whole session.
After today, you will have many fans.
Yeah, I'm your fan, and the other organizers. You're great. You are very strong people. You know. Thank you so much.